So today is January 20th, 2021. It is a historic day here in our country, the United States, because we are inaugurating Joe Biden as the 46th president of the United States. At noon today, Donald Trump will no longer be president of the country, and we will be swearing in Joe Biden as president and Kamala Harris as vice president. I really want to encourage you all to check out some of these proceedings as the day goes on. If you've seen any inaugurations in the past, this will look quite different than President Trump's inauguration 4 years ago or President Obama's inaugurations 8 and 12 years ago at this point. But it still represents a historic moment for our country as we kind of symbolize what democracy should be in the country. You'll see that there are a lot of security measures in place because of the recent attacks on the Capitol, which took place 2 weeks ago. You'll also see that there is social distancing in place because of COVID-19 pandemic. But you'll also see that members of both political parties, both of the major political parties here in this country, former presidents, celebrities, other formerly elected legislators are all coming together today to celebrate this transition of power. And regardless of what your politics are, this represents an important moment for our country, an important moment for us to think about what has happened over the last 4 years and even before that, and think about how we can impact positive change going forward. Certainly, there is a lot to reconcile because the last 4 years have been tumultuous, and that's to put it lightly. But I think that what we will see today is going to be symbolic of the fact that people of all backgrounds are willing to be a part of the solution. So check it out. You might find that it's at least interesting and at best inspiring. And I want you all to dedicate some time to doing that, but you should also dedicate some time today to biology and preparing for our first unit exam, which will take place tomorrow. Today, we are going to do some reviewing of the course that we've taken so far. So our objective is to cover the essential concepts and terms that are represented in this first unit of biology. I'm going to run through some of the slides that we've previously seen, so I'm going to go through them pretty quickly. Of course, this is being recorded, and you all will have access to these recordings if you want to go back and take a look at them, and I'll also give you access to these slides as well. But I don't want to spend too much time today, so you'll see me and hear me moving pretty quickly. And then we're going to do some more interactive work that will allow me to kind of cover some topics that perhaps you all are still struggling with. So I'm excited about that. The essential question, what do I need to know in order to master the content from Unit 1, Biological Macromolecules? So we will cover that today. This is the 6th day of our first unit. Tomorrow will be the 7th day, and we will take our exam. And then Friday, we'll have a day to decompress and for me to check in individually with each one of you, as well as to answer any questions you might have about outstanding assignments. And you all will have some time to work on those assignments as well. So let's go ahead and jump into it. So in this first unit of biology, we cover 3 essential standards. So this first unit is about 10% of the course, because we've got 30 essential standards that we will cover before the end of the class. And it was all about the biological macromolecules. We know at this point that there are 4 of them: carbs, lipids, proteins, and nucleic acids. The first standard is all about those 4 biomolecules. Again, we can call them various terms: organic molecules, biomolecules, organic polymers, whichever your preference is. You are more than welcome to refer to them in that way, as long as you know we're talking about those same 4 things. And here it is right here. Compare the structures and functions of the major biological molecules, carbohydrates, proteins, lipids, and nucleic acids, as related to the survival of living organisms. So they each have unique shapes. They're made of different atoms. They're made of different monomers. We have to know what those atoms and monomers are, but we also have to know what these biological compounds do and how they allow for living organisms to survive. The second standard is about how DNA codes for proteins. This is an important standard, but it's not one we focus too heavily on in the first unit because we dedicate a lot of time to it throughout the course, but primarily in the middle of the course with units 4, 5, and 6. And it says, summarize the relationship among DNA, proteins, and amino acids in carrying out the work of cells and how this is similar in all organisms. Okay, that's an important statement there. We know that all living organisms have to be able to carry genetic information, which they do in the form of DNA, and that genetic information is going to be translated into a new protein. And every protein gives us the traits that make us who we are. They give us our traits that allow us to reproduce, that allow us to digest foods and nutrients, that allow us to look unique. So we need DNA to carry that genetic information and then code for specific proteins. We'll talk a little more about that today as well. And then the last of the three standards in Unit 1 is enzyme specificity. Explain how enzymes act as catalysts for biological reactions. All right, so I'm going to run through this. We've seen this table before. Hopefully you all have written down this table, as I've asked you to do that several times. If you haven't written it down, at least hopefully you've taken a picture of it. And if you haven't done either of those things, you need to be tuned in and paying close attention right now so that you do have an opportunity to reveal some of this information to yourself. All right, so with our 4 biological compounds, as we can see in the column on the left, we need to remember 4 important things about each of them. We need to know what their monomer is, what the elements that make them up are, what that biological compound's function is, as well as some examples of the biological compound. And this will allow you to be able to compare and contrast the compounds, as well as to relate their structure and function to the survival of living organisms. Number 1, that first box. The monomer of a carbohydrate is called a monosaccharide. Monosaccharide, when we break that word down and we think about our prefix and suffix, it combines mono, which means one, with saccharide, which means sugar. So a monosaccharide is one sugar. When you combine one sugar with one sugar, you have a disaccharide. You have 2 sugars. All right, so a monomer is something that we combine with other monomers to build up a larger or to synthesize a larger structure. In this case, most of the time when we're talking about carbohydrates, we're talking about glucose. Okay, we're talking about the monosaccharide being glucose. The elements that make up carbs are carbon, hydrogen, and oxygen. And the cool thing about carbohydrates is that those elements appear in a 1 to 2 to 1 ratio. The function of a carbohydrate is short-term energy storage. All right, so when you eat something sugary, when you eat carbohydrates, you want to be able to burn that sugar off very, very quickly. We're not hoping to store that sugar for a long period of time. We're hoping for it to provide us quick energy. Additionally, carbohydrates provide some structure to specific types of cells, which we'll talk more about in the next unit. And then examples of carbohydrates. Most carbohydrates are sugars that end in the 3 letters OSE. And you can see that I've underlined those 3 letters in 3 different places here. Glucose, sucrose, fructose. There are other examples, including starch and glycogen as well. But again, when we think about creating mental cues for ourselves, we want to know and recognize that when we see those 3 letters at the end of a word, OSE, we should be talking about a carbohydrate. You got to have that mental cue. The next one, lipids. The monomer of a lipid is glycerol and 3 fatty acid tails. So it ends up getting the shape of a capital E, a backbone with 3 tails sticking out of it. The elements are the same, carbon, hydrogen, and oxygen. And keep in mind that we see those same 3 elements in all 4 of our biological compounds. All 4 of them have carbon, hydrogen, and oxygen. The function of lipids is to provide long-term energy storage. Okay, so when you eat something that is heavier in fats, that fat is giving you an opportunity to insulate parts of your body, including your cells, as well as to store energy for longer periods of time. Lipids also serve an extremely important function in cellular functioning because they make up the cellular membrane. We'll talk about that in the next unit as well. But the cellular membrane is kind of like a cell's skin. It's kind of what keeps everything on the inside. So lipids make up that skin. Examples of lipids include waxes, fats, phospholipids, some steroids, and oils. The next biological compound is called a nucleic acid. The monomer that makes up a nucleic acid is a nucleotide. And nucleotides are made up of three parts: a 5-carbon sugar called pentose. Again, there are those 3 letters, OSE. Pentose is a 5-carbon sugar, a phosphate group, and a nitrogen base. The atoms here, we've got 2 additional atoms that carbs and lipids do not have. We've got, again, those same 3, carbon, hydrogen, and oxygen, but we here add nitrogen and phosphorus. The function of a nucleic acid is to store genetic information, as well as to control all of the cell's activities, as well as to control production of proteins. So really, nucleic acids, you can make an argument, are the most important of these 4 biological macromolecules, because they really are the boss of all the other things. They determine which proteins get made. They determine how lipids get organized in the cell membrane. They determine what kind of signals are being sent out by carbohydrates. All right, so these are extremely important compounds, nucleic acids. They contain genetic info, they control which proteins are produced, and they also control all of the activities of the cell. Who gets to go where, who gets to go when, who gets to come in, who gets to go out, these things. And then there are only 2 main examples, DNA and RNA. DNA stands for deoxyribonucleic acid, and RNA stands for ribonucleic acid. And then lastly, we introduce proteins. The monomer of a protein is called an amino acid. We've got 20 amino acids that appear naturally. The elements that make up a protein, we've got those same 3, carbon, hydrogen, and oxygen. But here we also add nitrogen. The function of a protein is to really do everything else that the other 3 things don't do. They provide assistance to our immune system. They carry oxygen in the blood. They help to break down sugars. So some examples are enzymes, which we talked a lot about and we'll talk about today. Hormones, which are chemical messengers that are sent from your brain to other parts of your body or from your kidneys to other parts of your body. Insulin, which again helps break down sugars, and then antibodies, which help with your immune system. They recognize pathogens that are not supposed to be there and help to break those pathogens down. All right, so we did this mind map as well, which I'm just going to run through. In this stream, the first stream on the left, we've got proteins, which are made of amino acids. And examples include enzymes, hemoglobin, antibodies, hormones, and insulin. If at any point you want me to go back so you can take a quick screenshot, I'm happy to do that. But this is all stuff that we've seen and spent a lot more time on. So hopefully you guys have this copied down already. If you don't, it's not a big deal. Don't feel bad about it. I do want you to get this information regardless, but just let me know. That second stream, we've got carbohydrates, which are made of monosaccharides, also known as glucose or cellulose. In the third stream, lipids, which are made of glycerol and 3 fatty acids, whose jobs include long-term energy and insulation. And then lastly, nucleic acids, which are made of nucleotides. 2 main types are DNA and RNA, and that lead to genes or proteins, right? So your genes eventually code for your proteins. So this is a cool mind map that just gets you started to make the connection. between and within these different organic compounds. So take a screenshot of this if you don't have it written down, or a picture, whatever you need to do to get this information. Okay. Now that second standard, like I said, is the relationship between DNA and proteins. We won't talk too much about it in this unit. You will have a couple questions about it. What you need to know is that DNA contains the instructions to make proteins. I'm going to send that to the chat because that's a word, that's a bar. You all need to have that information written down. DNA controls, sorry, DNA contains the instructions to make proteins. And then lastly, proteins are made of amino acids. Please, just send a message to the chat. You can copy that down word for word in your notes, and it will be quite helpful to you. We've got that explained in more words on your screen. So nucleotides are arranged in a very specific order. And that order determines a specific DNA sequence. That sequence determines which amino acids are going to be put into place and linked together. And the order of those amino acids determines the specific shape and function of a protein. All right, so I love this slide. It really breaks it down quite well. I don't want to say too much more about this right now. We'll talk about it later in the course, but this is good information to have. This is good stuff to know. All right. And then lastly, we had a lot of information about enzymes. Historically, our students do struggle with the concept of enzymes, and enzymes come up quite a bit, not only on the Unit 1 exam, but also in the EOC exam that you all will take later in May. So we need to be sure that we have gotten as much practice and given as much thought as possible to this concept of enzymes. We talked about enzymes having three special qualities. Number one, they are catalysts. A catalyst is something that speeds a process up. So an enzyme being a catalyst means that it speeds up chemical reactions. Number two, enzymes are specific. So they only work with unique substrates. Not every enzyme is going to work with every substrate, just like not every key is going to open every door. An enzyme needs to find the substrate that has the compatible shape to fit with the enzyme's active site. Okay, so the active site is like the lock. Every lock, if you look on the inside of a lock, has a really specific shape. It has a cutout. The key comes in because the key fills in the cutout. And when the key is inserted in the right way and it has the correct shape, then it allows the door to be open. It unlocks the lock. Same thing with enzyme and substrate. The enzyme has an active site that has a specific shape, and the substrate also has the compatible shape to fit inside of that enzyme. Now, if I have some other random substrate that doesn't have the right shape, they're not going to fit together. All right, they've got to have compatible shapes, and this is what gives enzymes the quality of specificity. And lastly, enzymes are reusable. So an enzyme is going to be unchanged. It remains unchanged in a reaction. The same way you put it in is the same way it's going to come out. So we've seen this slide before. We saw it yesterday. We saw it Friday. You need to know those three things about enzymes: catalysts, specific, reusable. Okay. So enzymes as catalysts. What does that mean? Well, we talked about the fact that enzymes speed up reactions, and the way they do this is by lowering the activation energy. So we saw this same image yesterday. You can see that that green hill is much shorter than the red hill. And that's because the green hill represents a reaction that used an enzyme. The red hill represents a reaction that is not using an enzyme. Okay, so we need to be concerned about the fact that enzymes lower activation energy. They make a reaction easier to undergo. We talked about this as well. Enzymes help reactions that would already be happening naturally go faster. Okay, so the people on the left, they'll eventually be able to start that fire. But on the right, we can see that that fire is being started much easily and much faster, much more easily. And then I want you to take a look at this imaging that I put together. So the sentence says, as long as an enzyme shape is not changed, it can be used in reaction after reaction. And this is why we call it reusable. So you take a look at this. We've got an enzyme. We can see that that enzyme has an active site right here. So of course, it needs to fit with some type of substrate that is going to be similarly shaped, like this one, a heart. That's going to fit right into the enzyme's active site. Now, of course, we have more than one substrate. There are going to be substrates all over the place. A substrate is just some other chemical compound that the enzyme is going to act upon. So the substrate is going to go into the enzyme's active site, and that's where the action takes place. Let's take another look at that. The substrate goes into the active site. In this case, the enzyme is going to break the substrate down. So let's look at that. Substrate goes in, and enzyme breaks the substrate down, and now we end up with two new products. But of course, we still have a lot of substrate located throughout the rest of the cell. So this enzyme is going to move to a different place in the cell where it's going to be able to act on another substrate. And we've got this same process happening here. The substrate is going to go into the active site, and then we create new products. Okay, and this enzyme is going to continue to move all over the place throughout the cell, doing these same chemical reactions, breaking those substrates down and creating two new products. We talked about this slide yesterday as well. I wanted to recover it because I didn't know if you all fully understood what was happening here. But to put it in simple terms, we got to think about it this way. Let's think about it in terms of candy. It doesn't matter if I've got a Twix bar and I break that Twix bar in half. When I break that Twix bar in half, I don't create two Snickers bars. I still have Twix bars. They've just been now broken in half. Same thing. If I've got a Skittle and I chop it in half, I don't now create an M&M. So when I break something down, it gets broken down into its corresponding subunit. So I can't break a protein down into a nucleotide. I can't break a lipid down into a monosaccharide. That's not how that works. So if we look at number one, I've got an enzyme called protease breaking down a protein. Well, if I've got a protein and I break it down, that means that it's becoming an amino acid. I can only break down proteins into amino acids. If I've got amylase breaking down carbohydrates, I have to break those carbs down into monosaccharides. If I've got deoxyribonuclease, another enzyme breaking down nucleic acids, I can't break those down now into glycerol and 3 fatty acids. They're nucleic acids, so they're made up of nucleotides. So if I break it down, I'm creating nucleotides. And number four, if I've got an enzyme called lipase that's breaking down lipids, those lipids will be broken down into glycerol and 3 fatty acids. Notice that all of our enzymes here end in the letters ASE. So OSE is used to signify a sugar or a carbohydrate. ASE is used to signify an enzyme, which is a protein. So don't be confused about that. Typically, enzymes will end in ASE. Okay, we talked about this yesterday as well. If an enzyme is placed into an unfavorable environment, it will become what is called denatured. All enzymes prefer to be in a specific environment where the conditions can be controlled and regulated, where you don't have things that are fluctuating heavily and often. Enzymes prefer to be in stable environments with controlled pH and controlled temperature. So let's take a look at this enzyme graph. This enzyme graph is depicting how an enzyme functions based on different pH levels. This red curve is showing you the enzyme functions better and better and better until it gets to a certain point, and then it starts to function worse as the pH continues to increase. The point at which the enzyme is functioning the best is represented by the peak of the curve, the top of the curve. And in this case, that happens at a pH of 8. We call that the optimum pH. That's when the pH is at its best. That is the ideal environment, a pH of 8. But sometimes the enzyme is clearly not working at all. So in places where we don't even see the curve because the curve has now hit the x-axis, is now at a level of 0, that means that the enzyme isn't working. It's denatured. It's been so damaged by its environment that the active site has changed shapes, and we call that being denatured. In this curve, we can see that happens at a level just below about 4.8, 4.75. Anything below that, the enzyme is not going to work. Even if it's 4.5, enzyme won't work. Even if it's 4.6, enzyme's not going to work. And then we can also see anything above 10.5, the enzyme functioning is impeded as well. So we look at 10.5, even something like 11, the enzyme's not working. So the denatured pH is anything below 4.75 and anything above 10.5. I did that again. Dang it, I hate when I do that. Okay. One more curve, same thing. We can see that the enzyme is what am I trying to say? Performing better and better and better. Its function is increasing until a certain point. Then it hits a peak, and then the enzyme starts to perform worse. That peak in this case happens at an optimum temperature. So we find the highest point in the curve, we drop it down. This peak in this case happens at 40 degrees Celsius. And then we can also tell that anything higher than about 62 degrees or 63 degrees, the enzyme is not going to work. We can't even see the curve in this place. So anything greater than 62 degrees, enzyme won't work. All right. So at this point, I want to transition to another platform. Give me 1 second. We're going to be using Nearpod. I'm sure you all have used that in other classes. All right, so I'm going to share a link with you all to the chat. I need everybody to go ahead and navigate to that link. All you need to do is click on it. It should take you to a Nearpod. I'm going to, hopefully this works. Share this tab. It's not working. While you all join, I'm going to take attendance. So I see some people have been coming in and out. Hopefully, they're not upset if their attendance shows that they weren't here today. But thank you to all who have been here. Abraham and Alexander and Ardesia, Brian, Brian, Cindy, Christian, Frayland, Ayana, Jason, Kathy, Michaela, Naphtali, and Rudy. So again, hopefully all these other folks aren't upset. Gloria, I think, contacted me. Where's Brianna? Not here. Okay. So I've only got 5 people in the class so far, but we've got 14 people in our live session. So where are you guys? I've got 6 people. Thank you. Thank you to Abraham. Thank you to Alexander. Ardesia, where are you? Rion, where are you? Brian, where are you? Cindy, where are you? Christian, Fraylin, thank you. Ayana, thank you. Jason, thank you. If you're struggling or if the technology isn't working, let me know, but I expect everybody to be tuned in. Ah, okay. Stop sharing. Again, if you need me to, all you should have to do is click on the link that's in the chat to access this. And you're just moving through these questions that I have set up for you. Good. We've got 10 folks now. There are 13 of us in the chat. So we still have a few folks missing. Are you all able to access the quiz? There should be a quiz that's coming up. But it doesn't look like any of you have started it. So after you do the pairing,
No,
go ahead.
for us at first it wasn't showing because I started from the parent and it moves to quiz 8 questions and then after that shows us like at the end like the test results. Like it
So
keeps moving by.
it's not letting you actually do the quiz.
Yeah, it like keeps moving by itself right now. Now, now I see you score 0 out of 8.
Uh, so okay, what about now? What does it say now?
Quiz 8 questions.
And can you take it?
Yeah, I can take it.
Okay, yeah, do that.
All right.
Mine keeps skipping to the end.
What'd you say, Brian?
Said mine keeps skipping to the end of the test.
It keeps skipping to the test.
The end of the test.
Oh,
It
really?
goes for the test.
Let me see. Oh, I can see that someone has started it now. I think it was something that I was doing on my part that was messing it up. So after you do the pairing activity, it should take you to a quiz automatically. And yeah, it looks like you should be able to do it now. I think I was sharing the results with you all instead of actually allowing you to take it.
Okay, it's
And instead of clicking the arrows that are on the side of the screen, you've got to actually click the next button. There's a button that says next at the bottom that'll take you to the next question. So don't click on those arrows on the side of the screen because that'll take you to the next activity.
a dumb idea to talk.
Okay, folks, it is now 11:36, so you all are good to go and enjoy your lunch break. I appreciate you all being here today, and we will take our test tomorrow, so make sure you study up.